This is a production of Cornell University.
So as Don mentioned, I'm with the USDA. I am a research geneticist, and I specialize in abiotic stress physiology and genetics in grape. And grape is the topic of today. So grapes are one of the leading horticultural fruits across the globe. And they're good to eat, but their end products are also really important and lucrative. Juice, raisins, table grapes, and of course wine. The thing that limits grapevine is a narrow climatic band. The cultivated grapevine is a Mediterranean adapted species, and so if you look here, pictured on the map, I'm trying to get my orientation with which slides to look at. If you look, it is limited by a temperature range between 10 and 20 degrees Celsius, which captures the Mediterranean area, and then grapevine producing areas you're probably familiar with when it comes to wine: California, Chile, Argentina, South Africa, Australia. But we know that climate is changing, and there are new challenges emerging when it comes to grapevine production. So pictured here in red are grapevine regions that we currently have that we're expected to lose by 2050. The blue region, or those are in red. The blue regions are areas that we expect grapevine production to be able to expand into, typically because of a warming climate. So with climate change, we're expecting for winters to become slightly warmer, but we're also expecting to have altered weather patterns. And so those altered weather patterns do not remove the threat of cold temperatures and limitations to grapevine production, because I think we're all familiar with, if you were here in the last couple of years, the Polar Vortex, which is a very common climatic event, but one that we just haven't seen in a while. And so pictured here is a heat map showing that, that Polar Vortex. I believe this is 2 years ago. The temperatures dove down, caused a lot of damage in grapevines, both here in New York and as well as in the Midwest. And I was just at a conference where all of the producers and all the winemakers are cold climate grapevine producers, so coming from Scandinavia, Canada, that kind of thing. We had a meteorologist give a talk about the polar vortex and about climate change, and he did a really great job of explaining it. And so I thought I'd include that here just so everybody has an understanding about how that affects growing grapes in this region, growing grapes in areas where climate is not a Mediterranean. So the traditional climate has a very cold pole and very strong trade winds, which keep cold air during winter trapped at the pole. But as we experience warming and preferential warming in the poles, the trade winds get weaker, and then these polar troughs become more regular as cold air is able to slip out of the poles and dip down into areas where we were not traditionally experiencing such cold snaps. The flip side of that is there's also warming peaks that occur in contrast to that. So instead of a generally warmer winter, we're faced with a more erratic winter, where there are going to be episodes of much warmer temperature followed by episodes of much colder temperature. This wreaks a lot of havoc on grapevines, particularly when you're pushing the temperature envelope. Pictured down here on the bottom is another example of some of this weather phenomena as it affects horticultural crops. The bottom panel here is a picture of the Midwest of America in April of 2006. So this is like April 4th, this is April 22nd. So you can see the greening up. That's the trees and the horticultural crops all bursting, breaking bud. In this year, 2007, we experienced what was a polar vortex, although we experienced it in the spring. So you can see how far advanced the green was when you compare these two. And then after the polar vortex, or the spring freeze, you can see how much of that foliage died back. In 2007, I don't even know the billions of dollars that were lost in horticultural crops because of this event. So these events are expected to increase. But it's not always necessarily bad news. With climate change, there's also new opportunities. And I'm from the Upper Peninsula of Michigan, which is this peninsula up here. Most people don't realize that this is actually attached to Michigan. I'm from this corner here. I was very surprised to see that grapevine production is moving north in Michigan. So here along the coast of Michigan, there's a lot of wine trails, a lot of good hybrid and vinifera wines get made here. But now this little peninsula here, there's a winery there. And as you can see by the suitability map, there are some regions where they're projecting that it is more suitable for wine. I can tell you from the snowfall levels that this is not going to be a fun place to grow grapes. But we have to realize that there are challenges coming with climate change, and there's also opportunities. And so it's my job as a geneticist with studying cold hardiness in grapes to try to figure out the most efficient way to get us through the challenges and take advantage of the opportunities. If we look at New York, where we're all familiar with, picture on the left is the grape growing regions of New York, and picture on the right is the hardiness map put up by the USDA. You'll notice that there's a correlation between the dark purple areas on the left with the green areas on the right. That's because temperature is the primary limiting factor affecting production, agriculture production in general, but definitely grape production. As I said, the European grapevine is adapted to Mediterranean climates, so it likes it nice and hot in the summer and dry, and mild and cool in the winter. Because of that evolutionary history, it isn't adapted to deal with our winters, and particularly if you go further north or west and north from here, cannot deal with the winters. It is only cold hardy to around -20 to -25 C, which is -4 to -13 F. And I'll apologize right now, not all of my slides have C and F included, but this is the range we're looking at for cultivated grapevine. Our most hardy cultivars in this region are like Riesling and Cab Franc, Cab Sauv. They can survive to these temperatures. So that's sort of the backdrop against what my job is about. And the goals of my position are to understand and clarify what the physiology is and what the phenotype is of what we say cold hardiness. So what does that mean? I'm also supposed to identify wild and hybrid germplasm that we can use to advance grapevine breeding to make new varieties that are better adapted. I'm supposed to characterize the complex architecture of those, genetic architecture of those traits, and then assess genetic mapping populations in order to identify candidate genes that play major roles. So I have a number of tools that I can use. Most importantly is the USDA cold hardy grapevine germplasm, which if you don't know, we house in Geneva. It's about 1,300 different grapevine genotypes. Those are all cold hardy hybrids and wild grapevine species that can survive our winters. The other half of the grapevine germplasm is in Davis, California, where all of the cultivated varieties and cold sensitive varieties are kept. So we have a pretty amazing resource here, and I definitely stress you should check it out sometime. I perform a lot of physiological measurements of grapevine bud survival and phenology with both field and lab-based work. And I'm trying to employ next-generation DNA and RNA sequencing technologies to move it forward to identify the complexities behind these traits. So I've already kind of alluded to this, but wild vitis is very important to grapevine improvement. I listed here just a sampling of adaptive traits that we would really like to have in cultivated varieties that we can find in abundance in wild species. So we're talking about cold resistance today, but there's also differences in soil adaptability, rooting, pathogen resistance, ripening, differences in vine architecture, fruit flavors and nutrition, and then budburst and deacclimation, which I'll talk about a little bit today too.
And what species are we talking about as wild grapes?
These are the major wild species that we have in the cold hardy germplasm here. There are about 20 wild species in North America. And I'm picturing a few here and introducing their names, not expecting you to know them all, but just to give you a sampling of the diversity that we see when we look at wild species. So they're all sort of ringed around the outside with Vitis vinifera, Riesling in the center. Labrusca, Cinerea, Estivalis, Riparia, Rupestris, and Vulpina are all major species we have in the germplasm. Vitis amurensis is something from Eastern Asia that's used a lot in Russian and Chinese grape breeding efforts. And then, like I mentioned, vinifera is in the center. So there are roughly 20 species in North America. There's something like 40 in Asia. They're adapted to different abiotic and biotic stresses because of their evolutionary path and where they have adapted. I've pictured here 4 different distribution maps for Rupestris, Labrusca, Cinerea, and Riparia. The bright green is a county level distribution. So you can see that they differ quite a bit in how prevalent they are and what parts of our country they cover. As I mentioned, cultivated grape is cold tender. -20 to -25 is its limit, whereas cold hardy wild species can reach -35 to -40. Not much can go below -40 and survive. That's the ultimate temperature for water. So now we're going to talk a little bit about what do I mean by when I say cold hardiness and cold hardiness. It's a very complex trait. It depends on which tissue you're talking about. For this talk, we're going to be speaking mostly about dormant buds, but it also includes phloem and xylem, the lifeblood of the plant, as well as the cambial tissues. All of these tissues have to deal with freezing stress in our climate, and they may or may not use the same mechanisms. There are differences in the genes that contribute to cold hardiness. There's differences in the physiology, so whether or not you are able to export water or increase sugar concentration or modify with cryoproteins. All of this can be different in these different tissues. And there's also mechanical differences between the tissues and whether or not they can prevent the movement of ice. So you can have tissue barriers that block ice propagation in the plant. There's also multiple versions of cold hardiness depending on what season you're talking about. So I'm picturing here on the bottom is a timeline going from fall through to spring. So if you get a very hard, cold frost early in the fall, you can knock the vine down and do damage before it's ready for winter temperatures, and thus impede its ability to survive the rest of the winter. You can have massive cold events during the middle of winter, which simply surpass the defenses of the plant and kill the buds. Or if you have too warm of a spring, you can bud burst early, and then you're subject to frost damage, depending on, like I told you, those troughs and peaks in the weather can really affect the fall and spring of bud phenology. When it comes down to looking at the winter, there's two major groupings, whether or not you have a mild or cold winter, so the intensity of the winter, and then the stability of the winter, whether or not you have very punctuated swings in temperature or you have sustained cold temperatures. So today we're going to talk about of all the complexity I introduced, we're going to talk about two portions of it: midwinter bud hardiness and endo dormancy and bud burst. So on the left is the standard acclimation-deacclimation curve associated with plants going through the winter. In the fall, they react to changes in light and temperature, gaining cold hardiness and moving down this U. So they're acclimating ahead of freezing temperatures. During the middle of winter, they're at their most hardy. And then in the spring, as temperatures rise, they deacclimate at a rate that is faster than they acclimated. You can assess whether buds are surviving winter and where they are on this curve by cutting them and looking for damage. So here on the left is the compound bud of grape. The primary bud, secondary, and tertiary all are green. This bud survives, well, until we cut its head off. The center bud here has sustained damage in the primary bud, which is brown. And the third bud here is completely dead. So from a producer's point of view, you could get fruit off of this plant. You're going to get only vegetative growth off of this because the primary bud is the fruitful bud. And you don't have any vine growth, and you're probably going to have to replace your vine. Endo dormancy and bud burst is another topic we'll get into in a minute, but basically we assess the state of dormancy and the ability to grow by taking buds from the field and putting them into growing conditions, and that's what's pictured here on the right. The reason that I have these two traits up here is because they interact and overlap with one another. So throughout the acclimation and deacclimation period, you also have changes in dormancy going on in the background. And so I'm trying to decide, or trying to figure out, how interconnected these two different traits are with one another. Can we separate them and identify advanced germplasm for each of those traits?
Jason, normally the primary bud is the first to kill.
Mm-hmm.
It's the floral bud.
Yeah, it's the most sensitive, and the thought is that because the floral tissue has more water. But we're working on that. So here's my 5th grade drawing of what it's like to be a cell that experiences cold snaps. So we have a cell on the left, and that's all the good bits on the inside there. You have a spike of cold temperature, and from what we know, the cell tries to get rid of as much free water as possible because free water freezes. So you get ice crystallization between the cells, or perhaps just outside the plasma membrane. This sort of freezing is safe, and it occurs at around -5 degrees Celsius. It's called the HTE, or high temperature exotherm. Cells that have this ice form are under stress from that ice, but also from dehydration and membrane fusion. So there's a lot of processes going on when you have ice formation. As temperatures drop, the buds supercool, so they suppress their ability to freeze to a certain point. At some temperature, and this varies throughout the winter and by genotype, that intracellular water can no longer be kept from freezing, and it freezes. That signals the lethal event in the cell. So at that point, the bud is dead. What exactly is happening when that ice happens, we don't quite know. We're working on that too. But basically, there's a whole suite of tolerance and avoidance methods that are postulated to play a role in this. Differences in membranes, cryoprotective solutes, chaperones, sugars, suppression of ice crystal formation, tissue barriers, all of these are thought to play a role. We can evaluate where the buds are on that cold hardiness U using a method called DTA, differential thermal analysis. And what we do is we take the bud, here's the dormant bud, make an incision across the bud cushion, so here's a cross-section, keeping this tissue intact. We put it into these trays, stack the trays in a programmable freezer, then slowly freeze the tissue. As the temperatures are decreasing, when you hit the HTE, that safe freeze, you get a spike of water freezing here. This is a peak of heat, and we recognize that as a change in voltage across these plates. So that's the safe freezing. And then as you continue to drop in temperature, you hit the lethal events, and you see the buds dying here in these little spikes. So what we do is all winter long, we go out and collect tissue, put them in these chambers, record these peaks, average the peaks, and figure out what the 50% lethal point is, and use that as a method of measuring where the buds are on that U. So for the last 3 years, I've done this with 7 different species and about 33 different genotypes, and I'm not going to show you all of the traces because it's way too overwhelming. But basically, what it looks like is in the top right corner here. This is the minimum temperatures in the black jagged line, and here's Riesling and Concord. The solid lines are those LTEs, the lethal temperatures, and then the dashed line is just a trend line, so you can see that Concord is hardier than Riesling. This is something we know very well. So when we look across these 7 species and all these genotypes from hardy to tender, we basically come up with the result. Riparian amurensis are the most hardy varieties. Then the rest of the wild species are all within, there we go, all within about a degree of each other, Acivalis, Cinerea, Labrusca, Rupestris, and Vulpina. All of the wild species are hardier than the hybrid varieties, and those hybrids are hardier than vinifera. So just keep that in your mind, mostly the two ends of this. Riparian amurensis are very hardy, hybrid and vinifera are very tender. If we look at 2012, 2013, here I'm only showing you riparian amurensis and hybrids. You can see that we had this black dashed line is the freezing point, and these are minimum temperatures. So you can see we had this is a pretty mild winter. Temperatures go back and forth across the freezing point several times. You can see that hybrid amurensis and riparia are all about equally hardy, but way more hardy than temperatures demand. After this cold event, you can see a slight dip. They're responding to the cold temperatures. Then there's a warming event. Amurensis responds by deacclimating. And throughout the rest of the winter, you have this divergence. So this isn't a warm year. This is a cold year, 2013, 2014. You can see that the lines are further down. They're more hardy in this year. And now the blue line, which is the hybrid line, is the least hardy. Again, you have responses to temperature and deacclimation all the way through the season. Here's last year, which was also a very cold year, but it was a very different cold year. Instead of having lots of temperature swings throughout the winter, we had extremely cold temperatures for 2 months straight. So you can see that the lines are much less wavy, and this cold event here pushed all of the genotypes, even the sensitive hybrid varieties, far, far hardier than we had seen previously. So this is where the type of winter really matters. The last year's winter that we had pushed hybrid varieties way past what we would expect them to normally be able to do, simply because the right types of temperatures occurred. They were pushed down and kept down. They weren't allowed to deacclimate. So the type of winter determines the extent of bud cold hardiness as much as the genotype does. And here you can see I drew the dashed red line to indicate the hardiest point for riparia. And you can see that in the first year, the hardiest point is sensitive relative to the next 2 years. The point of this being is the buds do not supercool to maximum potential unless they have to, unless they're pushed to do it. So there has to be some sort of active part of this going on. Here are the other points for riparia. So you can just see that it gradually got more and more hardy across the 3 years. Assessing bud cold hardiness using LTE is location and year dependent. So this makes it really hard to use to track down genetic traits because it's always changing with the field. But I'm still tasked with figuring out how to breed for this. So what's the genetic architecture of this trait, and can we breed for it? To do that, I looked at Vitis riparia. So this is that North American, very adapted, cold hardy wild species. You can see here it has a huge distribution, and it goes well into Canada. We just don't track where it is in Canada. So I looked at 43 different varieties of riparia. And I'm going to go sort of fast through this part, just to show you the variation across those 43 different individuals. So here's 2013, 2014, cold year. And these are the LTEs for the different riparias. What you'll begin to notice is that they don't all follow the same pattern. So they're not all responding to temperature the same way. You've got some that are sensitive, some that are hardy, some that are reactive, some that are static. It's across the whole year, or the whole winter. And here's the most recent winter. And you have the same sort of trend. So even when everybody's pushed as hard as we can push them, you still get a massive amount of variation. Get to the end of this. Okay, so basically at any point in either one of the winters, there's 10 degrees Celsius range of variation. So we have a lot of stuff that we can look at for breeding material. And there's great potential for incorporating some of this new material into breeding programs that include riparia for grapevine breeding. The interesting thing is, northern varieties did not come out as always the most hardy. So in the first year here, varieties that were hardier than negative 32 degrees Celsius were from Montreal, Montana, Ontario, and Illinois. Illinois is considered south in this distribution. And in last year, we again had lots of northern material, but also Illinois, Kansas, Nebraska, Iowa, and Texas. So it's really interesting. This trait does not appear to be tied to geography. It seems to be locked within the species. Okay, so now we're going to move to the next trait, dormancy. Dormancy is a process that occurs throughout the growing season and throughout the dormant season. It starts during the growing season with paradormancy. This is where the growing tip is suppressing the growth of buds behind it. So this is what happens when you prune a plant and it gets really bushy. You release it from paradormancy. That's controlled by hormones. The next type of dormancy is endo dormancy, and this is what occurs in the fall. Light changes, cold temperatures drop, and the canes and vines ripen off. So they turn from green to brown, getting woody. That's called paraderm. It advances from the base of the cane to the tip of the cane, and as it travels, the buds that are covered in that paraderm are ready for winter. We don't know what keeps these buds in this state of endodormancy. It's something molecular metabolic. If you go out during the endodormant period and hack off a vine, stick it in water, and try to get the buds to grow, it won't burst. There's something keeping those buds silent. After you successfully complete endodormancy, you move on to ecodormancy, and this is a state where the temperatures are keeping the plant dormant. So if you go out and cut a vine in ecodormant stage and put it in a hot glass of water, it'll grow. So this is an environmental form of dormancy. After it gets warm enough and growth resumes, the plant goes back into the growth season. But if it gets there too fast and we have a frost event, then this happens and we lose the fruit-producing parts of the vine. So that's why this is related to cold hardiness. The way the dormancy clock works is the buds somehow keep track of the amount of time that they spend between about 0 degrees Celsius and 7 degrees Celsius. There's an optimum area there. They count the hours throughout the whole winter. And that's what's depicted here. The two winters, 2012 and 2013, so warm versus cold, in this green shading is that sweet spot for that dormancy clock. So the buds are tracking temperatures throughout the winter, and this tells them when it's safe to burst in the spring. So I zoomed in here just in the yellow box on the same dates across those 2 years, the warm winter and the cool winter. And if we imagine that we want to get to 12 o'clock noon before we burst and look at this region, you can see that in the warm winter, they made it to 7 o'clock in this chunk of time. And the next year when it was cold, they only made it to 3. What that means is you need much less warm on the other side of this bracketed area to burst. And in fact, in this year, you can see that they've got plenty of chilling. The buds burst and we got frost damage. And this year, this same amount of chilling didn't mean the same amount to the buds because the clock was slower. So it's really important that you grow things in regions that have, say, a clock of 12, that you don't grow things that are going to be supercharged in that time. This is what the chilling hour / dormancy clock looks like for across the country. I'm not sure if you can read these numbers or not, but it starts with 0 in Florida. It never gets below 7. So plants that would grow in Florida wouldn't need chilling in order to fill their dormancy. You gradually gain more dormancy as you move up the country, along the coast where it's cool, up in the Pacific Northwest where it's cool, and then up in the northern plains, there's actually very few chilling hours. And this is because their temperature swings are so dramatic. They go through from summer to winter so quickly that that sweet zone is actually very tiny. So that's chilling hours. And we can evaluate what the chilling hour requirement is for different species, for different genotypes by a simple test, collecting the vines, putting them into cold storage in that sweet spot, and then testing how fast they burst. And the data looks something like this. This is Cabernet Franc, Cab Sauv, and Riesling. At the start of the experiment, you can see that they take quite a few days to burst, but as you add chilling, they get faster and faster and faster. If you take a given set of hours, at 1,000 hours here, you can see that Cab Franc and Riesling are fully chilled and they're bursting really quickly, and Cab Sauv is lagging behind. One of the consequences of not being fully chilled is that you have desynchronized bud burst. That leads to desynchronized flowering and desynchronized fruit ripening. So this is not a good trait to have. You can see here, this is Riesling that's fully chilled. This is Cab Sauv that's not fully chilled. The buds are bursting at different rates. So if we look across a wild grapevine, these are the trends in what the species do when we're faced with chilling hours. I want you to notice that all the species respond in the same way to chilling. They all need chilling to burst, but they have the same reaction to the same amount of chilling, but they start at different spots on the y-intercept. And so different species and genotypes have different chilling requirements. There's low chill, rapid bud bursting species, moderate species, and then high chill, slow species. These tend to separate along a northern-southern gradient as far as distributions go. So it's very interesting. We might have an adaptive trait in this data set. So we can select specific germplasm to breed for these different traits. So if we look at this in Riparia, again, we could look at this with 43 lines. I'm not going to show you 43 lines. I'm just going to show you a standard Riparia line. So this is Riparia's response to increasing chilling. It gets faster and faster for bud burst. So chilling hours are going up here, bud burst days on the left. This is the maximal range of variation within the 43 Riparias. Okay, so all of the Riparia lines fall between this blue and this green line. What we see is that the steeper the slope is, that indicates that that genotype responds more to a given unit of chilling. And the position on the y-axis indicates the Endo Dormancy barrier. So the higher you are on the y-axis, the more dormant you are through the winter. If we look at, if we imagine that we breed with these Riparias and then put the hybrids out into North America where we're growing hybrid grapes, pictured here is North Dakota, Nebraska, and New York, and the chilling hours that you typically receive in those regions. So you can see, depending if you planted for example, the green variety in all three locations, you have very different bud burst rates because of their chilling hours and seasons are different. If you add climate change and winter warming to this, you shift these all to the side. And so when you're thinking about breeding for cold hardiness from a dormancy point of view, you have to be thinking way ahead to the shift in climate, this increase in chilling hours. And so now you can see it's even more important which varieties you choose, because the faster you break bud, the more susceptible to frost you are. And this is the 43 different genotypes of Riparia. Yellow is southern. I've called them southern. Blue is northern. In the two columns is the average LTE for the 2 years I showed you. You can see that there is no apparent trend between darker red with northern or southern. The LTE trait is sort of shared across all of those individuals. If you then include the dormancy information as slope and y-intercept, so here's what slope and y-intercept means. Bluer in slope means you react stronger to the chilling. Blue in the y-intercept means you have a higher endodormancy. You can see there seems to be a possible association between the southern having higher endodormancy and a higher response to chilling. In the north, there's more red, so you tend to have lower endodormancy. And so there's several potential varieties, if you look across these traits, that might be good for breeding. So we'd want red in the slope, blue in the y-intercept, and then also hardy in the LTE. And so I've highlighted a few of them here. And you can see this is from Missouri, Illinois, Iowa, Kansas, Montana, and Ontario. So you can see that you have the option to breed with far more material if you consider southern Riparia in a grapevine breeding program. Okay, so now we're going to do a quick shift out. So this is the phenotyping effort that I've been leading in Geneva, trying to figure out these traits, trying to figure out how much of it we can use in grapevine breeding. But there's only so much you can do in the field because, as I showed you, every winter changes. And it gets very frustrating to try to run a controlled experiment when the outside keeps changing on you. So what we do is we take the field research to the lab. And we can take hints from the field research. So we know that all the species react the same to chilling. increasing both their bud burst rate and their synchronicity. The Endo Dormancy and LTE traits seem to be a little bit tied to one another, but maybe that's only by chance. And there's variation within Riparian suggesting that Endo Dormancy may be adaptive and LTE may be a species static trait. So then I want to know, can we determine the mechanism behind these traits? And so I'm going to introduce my student, Al Kowaleski. He is working with me, and we're working on trying to dissect deacclimation. So we want to look for the correlation of deacclimation phenotype and bud burst phenotype. So what we did is we took Vitis amurensis, Vitis riparia, those are those cold hardy wild species, and Vitis vinifera, and we subjected them to a test. And so here are some of the stats on the species. Amurensis and riparia are fast deacclimators, limited chilling hours, and fast bud bursting species. In vinifera, we have slower deacclimation, slower bud burst, and if you look at Riesling and Cabernet Sauvignon, you'll see that there's a pretty big difference there in the chilling hours. So we can compare the normal bud burst rate with a delayed bud burst rate. And what we do is we take a whole bunch of canes from the field, put them into growing conditions where everything is controlled, and sample the material for bud hardiness, check it for bud burst phenology, and then look at gene expression. So here's an example of what the experiment looks like on day 10. If you can see, hopefully you can see that amurensis has green swollen buds. So I mentioned it's the fastest bud breaker. If you look at riparia, Riesling, or Cab Sauv, and you can find the buds, they're not breaking bud. So this is just an example of what our data looks like as we're taking it. We have several hypotheses associated with the gene expression when it comes to the processes of deacclimation, bud burst, and cold hardiness. So based on the field data, we would think that deacclimation genes would be on in all genotypes, but would be tied to the actual rate of deacclimation for each species. Bud burst genes should be all upregulated and also tied to the rate of bud burst for each species. And that genes that are important for cold hardiness and cold survival should be massively downregulated because there is no longer a stress. So what we did is we collected hundreds of cuttings on February 2nd, and here's the chilling hours in the field on February 7th. It's about 750 hours. And what that means is that amurensis, riparia, and Riesling are all fully chilled. Cab Sauv is not. Here's their field deacclimation response. This is from LTE, so we know that riparia and amurensis are fast. Riesling and Cab Sauv are slow. And their bud burst phenology in this experiment reflected their chilling hours. So we had amurensis first, riparia second, Riesling third, and Cabernet started bursting about the same time as Riesling, but it was really stretched out because it was not fully chilled, so not synchronized. This data is showing you the deacclimation rate at 22 degrees for riparia and amurensis. So these data points are the cold hardiness levels measured by LTE each day. And then here, in these dashed lines, is the temperature of some control canes we left in the cold room at 4. So treated exactly the same, but kept at 4 degrees instead of 22. If we look at cultivated grape, here's the deacclimation rates at 22C and at 4C. And the first thing you'll notice is that the rate of deacclimation of ecodormic buds is exactly the same. The slope is not different amongst these 4 varieties. So this is totally in contrast to what we see in the field. And what we think is this happened because we put them into 22 degrees Celsius 24 hours a day, which is not a realistic situation. This is the problem with going from the field to the lab. Sometimes you overshoot. So we basically put them in a maximum growth rate condition, and they all deacclimated the same. However, at 4 degrees C, you see that only amurensis was deacclimating at a different rate. So this means that that species is able to respond to much cooler temperatures. And this would make sense from a field point of view because it deacclimates fastest in the field as well. And you spend much more time at 4 than you do at 22. So in this experiment, we decoupled deacclimation and bud burst. So we can't look at gene expression patterns in the same way that we were planning, but we can still look at what was going on. So here are days 0 through D8. This is the deacclimation rate. These are the days that we sampled for RNA-seq. Genes that have shared expression that are important for deacclimation are going to be about the same for all 4 varieties because they're all deacclimating at the same rate. Genes that have a staggered expression pattern are going to be tied to bud burst because they're bud bursting at different rates. And genes that go down are related to cold hardiness. So these are our assumptions. So what we did is we collected material on each of these days for the entire experiment, ground them up, and did extractions for RNA, and then sequenced that RNA. And I'm not sure how many people have done RNA-seq in here, But basically, we sequenced over almost 1 billion RNA sequencing reads. So it's an enormous data set. After we cleaned it up, we have about 500,000 reads in this experiment. If you take those 500,000 reads and align them to the genome and try to figure out which genes are turned on, which genes are turned off, out of the 32,000 predicted cultivated grape genes, we picked up about 13 to 16,000. So you go from almost 1 billion points of data to hopefully just 13 to 16,000, which is still a massive data set. If you then apply some statistics and look at significantly differentially expressed genes, we can cut that data set way down to the max 4,000 genes in Vitis amurensis are on or off in this experiment, and in Rhiparia, just under 2,000. So it's still a massive data set to deal with. But this is how we're going about looking at it. If you're unfamiliar with microchip analysis or RNA-seq analysis, this is what a heat map of RNA-seq data looks like. Blue to red indicates down to up gene regulation. So blue genes are downregulated, red genes are upregulated. And what you can see here in this cluster and this cluster is that the genes start out downregulated and gradually increase in gene expression. And the difference between these two is this one, I guess you could say it's gradually upregulated, whereas this is early upregulated. You see the transition from blue to red is faster. In this box and this box are downregulated genes, so they start high and they end low. And then in this little box here, you can see they start blue, go red, go back to blue. So these are genes that are coming on, probably starting a signal cascade and then being shut off. So those are peaking genes. So again, these are our gene numbers we have. This is amurensis I'm showing you. So this is the 3,923 genes. Each line is a gene. If we look at upregulated genes that are found in all 4 of our different genotypes, there's only 447 genes. So this is much more manageable than even 3,000 genes. If we look at genes that are shared in the downregulation category, there's roughly 230 across all 4 varieties. So now we can look at these genes and see what sort of differences in gene expression are going on in all of these processes. So I picked some just at random. This is not, I didn't choose this because there's a ta-da moment coming or anything like that. I just wanted to show you some of the gene expression patterns and some of the processes that we're identifying that are related to, in this case, deacclimation. So as I mentioned, deacclimation occurred equally among all 4 varieties, so we'd expect gene expression to be more or less all up for everybody, and you can see some of that here. Gibberellin, 3-beta hydroxylase, OX1, these are things related to hormones, gibberellin and auxin. ABC transporters, lipid transporter proteins. I'm not sure if you can see the y-axis here, but it goes from something like 2,000 to around 18,000 expression units, so massively upregulated lipid transfer proteins. Histone deacetylase is important for actually shutting genes off. It ties up the DNA into histones, and so this is interesting that they're upregulated. And then cupin here, which is, cupins have been seen to play a role in germination in wheat. So a lot of this makes sense in a deacclimation process, in a process that's, the bud is getting ready for growth. And a picture over here is a summary. Gibberellins, auxins, lipids, like we said. I'm sorry, the colors of the lines here are indicated by the genotypes in the same color. But if you want an easy key, blue and green are wild, orange and purple are cultivated. So genes involved in budburst, we would hypothesize that these would be turned on fastest in amurensis because it bursts fastest, followed by Rhiparia and then the vinifera's. And you can kind of see from this that we actually need to extend our experiment beyond day 8 because we're not picking up a lot of that cultivated response. But here are 6 genes that are upregulated rapidly and we think could be playing a role with budburst. TSO1 is a floral organ differentiating gene, so this makes sense. The floral meristem is probably becoming activated. GSA4 has a role in gibberellin. Ethylene has a role, alpha-tubulin and alpha extension. These are both expansion and elongation type genes, and auxin response factor. So a lot of growth things. If we look at downregulated genes, here's another sample of just 6. This plexstringy or pH domain protein, that's the only information we have on it, but it goes from something like 14,000 down to 2,000, so massively downregulated. This has a role in membrane transports. SRC2 has been picked up in soybeans in response to cold regulation, so this makes sense, another good candidate. Dreb genes and Werke genes are both transcription factors that play a role in cold hardiness, dehydration stress, even some of the other stresses depending on the family member. Galactanol synthase plays a role in getting rid of raffinose, and raffinose is a sugar that's been seen to be upregulated in response to cold, so this makes a lot of sense. It's being shut off. As with any gene expression study, you get a whole bunch of genes that have no function attached to them, unknown proteins or no-hit proteins, and some of them have really cool gene expression patterns. So here on the top are 2 that are downregulated quite a bit, 2 on the bottom are upregulated. So these are genes that we don't know what they do, but they clearly play a role in this process. They're picked up in all of our samples. And then there's a whole category of stuff that just has weird gene expression patterns and we can't make a lot of sense of. So you have to take it all with a little bit of salt. I've shown you a little bit out of that 450 and 230, but what we're doing is trying to figure out candidate genes and pathways that play a role in these processes. And so that's the ultimate goal. Can we identify pathways that are overrepresented within this data set? So we could then say, the deacclimation pathway is this. And this is what you get to work with if you're looking for doing pathway analysis, which is why I'm not going to show you today because I can't do this. But this is the metabolic pathway in grape for everything. It's not just cold response or anything. So instead, I did one pathway I zeroed in on because a lot of the candidate genes I saw in the data set were picking up in my memory about flavonoid biosynthesis. When I was a grad student and I had to do my preliminary examination, I had to write a grant on flavonoid biosynthesis. So it finally paid off. And so I saw that there's lots of these genes that I recognized, and so I thought I'd remake the pathway just to see what was going on. And so here's the pathway that leads from phenylalanine to flavonoids or to monolignals. And they all go through this sort of intermediate, which is really important. So if you go to the left of this, you turn into a flavonoid. If you go to the right, you turn into a lignol. These are the genes, and it's really not important that you know what their names are, but maybe you know chalcone synthase. I think that plays a lot of role in flower color and that kind of stuff, and so this pathway would lead to color and anthocyanins. This is peroxidase, just in case you're interested. So basically we have flavonoids or monolignals. These are the genes in that pathway. I picked up almost every single gene differentially expressed in all 4 genotypes in this pathway, but you can see that it's not always the same. So shared expression down, Amurensis and Rhiparia are starting to pick up. Pretty much shared expression across the lignols, but when you get to the flavonoids, Amurensis is way up and Rhiparia is way up. So this suggests that maybe in Rhiparia and Amurensis, the flavonoid pathway is playing a bigger role in getting those buds ready for budburst or deacclimation or something. Even though you see all the genes are up here, the expression level of this isoform is much lower than this one. So it just gives us an idea of what might be going on. And so here's that whole pathway. Pink is down-regulated, green is up-regulated, and dark green is massively up-regulated. So you can kind of get an idea of maybe some of the pathway that's playing a role in this. And my hope would be to take all of the genes, not just the few that I showed you, but apply that to some pathway analysis and then determine if there are other aspects of the metabolism that are unique to different species or unique to perhaps the difference between Cab Sauv and Riesling. So I've given you sort of like a buzz through physiology and phenotyping and next-gen analysis. And we've got a lot of future plans with this sort of research. We want to look at more controlled freezing experiments where we take all the canes into the cooler in the beginning of the season and subject them to freezing and warming in a controlled manner so we can sort of see their response to temperature better. We want to look at both endodormant and ecodormant deacclimation. What we looked at today was ecodormant. We also want to do some diurnal sampling. So, and Al's not particularly happy with this, but we're going to go out in the field and we're going to be sampling like every 4 hours over several days to try to get how the genes are being turned on and off as they warm up during the day and then get cold at night. Try to get a better handle on how much gene expression actually occurs near zero. Ultimately, we'd like to move this to association transcriptomics and look within mapping populations to try to find EQTL associated with dormancy, associated with deacclimation. And one area that's massively understudied is cold hardiness in trunks and canes. That's something we haven't touched at all here, and it's not something that I've done much work in. But ultimately, we'd like to move into that area to try to get a whole vine response. So with that, I would just say thank you to the people at USDA who support this work. My technician is Kathleen Deese, and a previous technician, Jacqueline Lillis. as well as a farm crew, Bill, John, Bob, Greg, and Nancy. At Cornell, of course, my student, Al Kowaleski, he's been helping out a ton and is going to be helping out a lot more. Bruce Reich is his co-advisor, and Tim Martinson and his team, Bill Wilsey, Steve Luce, Beth Takis, and Lynn Johnson is with the Cornell Statistics Group. She helps me out a lot. And then I've got some other collaborators on here. Oh, you can't even see it. That thing's in the way. Ann Fennell, Dan Chitwood, Michaela Centenari, and Grant Kramer. These people all play a role in these same sorts of phenotypes, and so we all kind of work together. And I will just ask if there's any questions. I do my best to answer them. Thanks.
This has been a production of Cornell University, on the web at cornell.edu.